Hi there, this is Charlie Destries coming to you with another video, and this time it's a video to discuss a particular technology with the goal of asking for your opinion on a survey that I put together at a site called thinkbigscience.com. Anyway, I'm working with a company in Sweden called Nanoxis, Nanoxis. Great group of people. They have a tremendous technology that's very simple. And sometimes the simpler it is, it's easier to overlook, in particular if you're walking around at a trade show, you're looking for those fancy $50,000 boxes with, cool interfaces and gizmos that do this and that and whatever. But this is, again, something that's relatively almost like, gee, why didn't I think of that when it comes to studying membrane proteins? And, what I did was I did some rather unpolished drawing and to discuss exactly what I'm talking about. So for you folks that are involved in proteomics and you're looking at cell membranes and proteins that are on the surface of those cell membranes for vaccine discovery and development, for drug discovery, and a bunch of other reasons, then this is a technology you want to pay a lot of attention to. And again, I'd love your opinion. So, generally speaking, what we're doing is we're looking at a cell. This is I hired the best graphic artist to do this. You're looking at a cell, and of course, inside the cell, you've got a ton of stuff, proteins, and when you lyse that cell and you try to get to this surface, to the proteins that are embedded in the cell membrane itself, it's difficult to really separate. You've got to do some fancy dancing to do that, and at times it's a mess. It takes a long time, and at the end of the day, you don't really have some really easy way to get to these proteins. Worst of all, you denature them, and so they're not in their native form. What if there was a way to take this cell, get rid of the stuff on the inside, but maintain the conformational integrity of these proteins so that you could actually experiment and interrogate what's on the surface of a membrane without breaking it up? Now, wouldn't that be cool? Well, essentially what this little company has done is they've taken a device. Well, here's their one-channel device. It's about the size of a microplate. And so you inject the materials in here, and it sits on the inside, and you can do some neat stuff. What you do is you take cells, and you create cell vesicles, basically like a ghost cell, but with all the intact proteins on the surface. And so this device then has this channel sitting in here. You inject the cell vesicles in there. The surface has a proprietary coating that essentially takes these cells, and again, these could be bacteria cells, they could be eukaryotic, prokaryotic, and it basically holds on to them. What's the word I'm looking for? It immobilizes them. So this is essentially the diagram of what I'm talking about. Here's the cross-section of that device that I just showed you. So we're kind of looking at it like this. And so on the bottom and the top of this channel, you've got these cells, these cell vesicles, I should say, that have intact native proteins that are sitting on the surface, essentially where you want it. So now you've got this immobilized cell vesicles with these intact native proteins, and you can add reagents through this. So just imagine, for example, and this is a blown up, this is essentially another graphic artist rendition, a blown up version of the cells that you just saw on the surface of this device. So imagine you've got this surface, you could put reagents through here, and let's say you put a protease in here first, protease number 1, which looks for a particular sequence in the protein, and so it's going to clip off those particular peptides. Boom! You put this protease through here, and what elutes is your peptides of interest, and you can put that into some further chromatography or detection device, LCMS, fluorescent-based stuff, whatever it is you're doing, and you can analyze. But remember, now you've got real pure membrane proteins that are sitting here. So just imagine, you can put one protease after another. You can slow it down. You can speed it up. You can shave the surface of the membrane, which is a terminology that's being used out there. And then think of other types of reagents, small molecules, antibodies, and so on. So the trick in this very, very simple device is that you have a consumable where you inject your cell vesicles of interest, then you inject your reagents of interest. On the effluent side, you essentially collect that, put it into whatever next phase it is that you do, whether it's more chromatography detection, whatever it is, and you're in business. Very, very elegant, simple, inexpensive, and high impact. Right now, there are several companies, biopharmaceutical companies, that you would recognize the name right away of, that are moving their research programs forward because of this technology. Now, this little company, Nanoxis, is recognizing that they've got something pretty hot. So they're out there essentially talking to the investment community in Europe and North America and Asia, and they are looking to build up a war chest to bring this out to the marketplace in a big way. But they need some information, and they need your opinion on some applications for this particular area, whether you're involved in the vaccine, therapeutic, either prophylactic or therapeutic development, monoclonal antibody therapeutics, or if you're a proteomics researcher doing some top-down or bottom-up stuff that you're looking for a way to really find ways to really grab the membrane protein itself in its native form, then you want to take a look at this. The company's called Nanoxis. My survey is at thinkbigscience.com, right there. And we want your opinion. It's only 5 minutes long. It's about 20 questions. And we'd love to hear from you and let us know what you think about this. And you know, the good news and the bad news. You know, gee, this is a good idea. No, that's not a good idea. That's all we need from you. And so we need it as soon as you can get to it. It's a lot of fun. I think you'll enjoy it. And more than anything, you'll be one step ahead in the knowledge base of proteomic scientists. Hey, this is Charlie Destries. I enjoy talking to you. Let me know if I can be of any help to you. And we'll talk real soon. Bye now.